Okay, we're going to move on to something which is somewhat different because it's more akin to what we heard before lunch in the context of electron transfer or electron tunneling. But I want to emphasize what we're discussing now are thermally activated processes. So there is something fundamentally different from a system where you, either with ambient or laser-induced light, are both heating up the system as well as exciting your electronic and vibrational modes. So we have to think about how this is all going to happen at KT. Now, enzymes are the basic catalysts in nature. They carry out reactions that are upward of 10 to the 26-fold faster than their comparative reactions in solution. And the question is, how do they do it? How do we then use the principles that we learn about enzymes, perhaps, to do de novo design, either in the computer or in the laboratory, of equivalent catalysts? And no one has come close, and the question is why. So that's what drives my research. But in the process, we've undergone this cross encounter with quantum mechanics, which in the end has taught us a great deal about how enzymes work. So our focus is a little different. Nature does use quantum mechanics, and the question we can discuss afterwards is, what does this mean? Is quantum mechanics driving the process, or is nature driving the process and quantum mechanics is coming along for the ride? So please keep that in mind. Okay. So there are two issues. One, as I say, you've had lots of time to read it, experimental verification. Why should you believe me when I talk about hydrogen tunneling at room temperature under the conditions of biology? And the second is the relationship of the observation of room temperature hydrogen tunneling to evolved function. Okay, so experimental verification. Now, we've heard all about electrons, and when we started this work, people were very certain, I think, that electrons could move quantum mechanically over long distances. And when we move to CH activation and hydrogen transfer, you have to take into account that the hydrogen is about 2,000-fold heavier than the electron. And so where's the dividing point for particles that will move quantum mechanically or not at room temperature? And you can actually calculate a de Broglie wavelength, and these data are shown underneath the masses. But the real issue is the mass of the particle in relation to the process that's being studied. And we know that for electron transfer, you have donors and acceptors that can be very far apart. We've seen up to 40 angstroms apart. And so the long wavelength of the electron is in the range of the distance between the donor and acceptor. Now, for hydrogen transfer, actually, it's pretty similar. Yes, the de Broglie wavelength is about an angstrom, but anyone who studies reactions in condensed phase knows that when you transfer a bond from one atom to another, the distance over which the bond will move is about an angstrom. So once again, the estimated de Broglie wavelength is in the same ballpark as the process that you're studying. And therefore, in that context, it's not surprising that tunneling is going to occur with hydrogen, protium hydride, free radical, hydrogen atom, you'll see. Okay. Well, that's great. That's a thought experiment. But how do we prove it? I'm an experimentalist, and the question is, what are the data that will support this? We've heard a lot about electron transfer. We've seen the temperature dependencies that led to the original proposal, at least, of low temperature electron tunneling. The distance dependence at room temperature is extremely important because it really validates the process as well as the models that describe it. Now, what about hydrogen? How do we verify experimentally that tunneling is taking place? Okay. Now, we have an advantage with hydrogen because we have isotopes. We have protium, deuterium, and tritium. And this has been used in many experiments to look at mechanism and try to understand reactions in condensed phase. And historically, the focus is on the ground state, where you have different vibrational modes. You have the same electronic potential energy surface, but because of the difference in mass, you have different ground state vibrational modes, where protium is higher than deuterium is higher than tritium. And when people then study these reactions across various temperatures, what happens when you go to increasingly high temperatures is that the spacing among these three isotopes gets smaller and smaller. So in this simple model, by the time you get to the transition state, there is no difference among the isotopes. And all of the isotope effect, these are kinetic effects, comes from the difference in the zero-point energies. And this leads to the kind of Arrhenius plot I show here, where you just look at the temperature dependence in an experimentally accessible range of protium and deuterium. You extrapolate back, and the lines will converge on the y-axis at the same point. There's no isotope dependence. Now, it has a lot of implications, all of which have been violated in biology. And I think this is important, because this is what's in the textbook. This is where we started. Now, this model predicts an isotope effect of 7 if you compare protium to deuterium. We now have reported isotope effects at room temperature of 700. Okay. It predicts that when you do a temperature dependence, as I said, the lines will converge back on the y-axis, but we actually get apparent intercepts of around 700, the size of the isotope effect. We have 3 isotopes. We can compare them. We can compare H to D and see how that compares H to T. These are related exponentially. There are predictions. They've been violated over and over again in enzyme systems. Furthermore, this simple kind of picture predicts that when you go to infinite temperature, your rate is limited by the time of a molecular vibration, 10 to the 13 per second. But when we do these kinds of analyses, we see Arrhenius prefactors of 10 to the 23rd. Okay, anomalies all over the place. What's going on? So prior to about 1999, this was the semi-classical expectation I've just shown you, where things will converge on the y-axis, so all the effect can be explained through zero-point energy, and the isotope effect will be about 7. Now, there are many, many theoreticians and experimentalists who have recognized that tunneling can occur, maybe to a small extent at room temperature. There's something called the Bell correction. Bell wrote, who was a lecturer at Cambridge or Oxford, I've forgotten which, and wrote a wonderful series of books explaining that, in fact, even in this semi-classical picture, the barrier gets more narrow near the top, and you start moving across the barrier rather than over the top of the barrier. This is referred to as a correction. It's also the basis for variational transition state theory, which Don Trular and many computational chemists have promoted. Now, this says that the isotope effect will be around 10, maybe. You get some additional discrepancy between the light and heavy isotope because the protium with the longer wavelength starts to tunnel prior to deuterium, which has to almost get to the top of the barrier. And this also changes the slopes of these lines, because now you have to put in extra energy for deuterium to get you closer to the top of the barrier, and the lines will appear to cross. Okay, all this is in the textbooks. Now, what do we see for enzymes? Now, this is a pattern I'm going to show you today, but it's been seen over and over again in many systems. It's not dependent on the nature of the reaction, the cofactor that's present, or whether hydrogen is being transferred as proton hydride or hydrogen atom. What do we see? Native enzymes, where we are restricted in our temperature regime to somewhere between, let's say, 5 degrees, and maybe we can go as far as 50 degrees, so it's a limited temperature range. But we see slopes that are equivalent for transferring protium and deuterium. Point 1. 2, as we start to modify the enzyme, introduce perturbations largely through mutagenesis or going to the wrong temperature regime for thermophilic proteins, etc., we start to see behavior that looks like semi-classical behavior, right? Seems to be behaving as it would, as I showed you early on, from zero-point effects. And then when we go to extreme impairment, we see what has been described as the Bell correction. Now, this has been seen kinetically, and we have lots of ground state characterization of proteins as well as we go from native to impaired enzymes, which show that, if anything, as we go in this direction to this direction, the distance between the donor and acceptor is moving apart. So it's not like we get somehow into a situation where they move together and we have more of a Bell correction. This is, in fact, these are, in fact, the data. Okay. So what is going on here, and how do we explain it? Now, many of you, or most of you perhaps, have not thought about temperature dependence of KIEs. I'm trying to make this as clear as I can. But as I said last night, if what I'm saying is really incomprehensible, raise your hand and I'll try again, because I really want to get this point through. So basically, we need a new theory that will explain the native enzyme with these parallel lines. How can the enthalpies be independent of the isotope? And then we have to explain a trend where we go through moderate to extensive impairment. So I'm going to give you some data first, and then I'll give you a model. It's always good to see some data. This enzyme is lipoxygenase. It's widespread. It's very important in physiology. It's been extensively studied because it controls all kinds of signaling events in cells. There are many crystal structures, and I'm showing you a structure from an enzyme from soybean. The center of the enzyme has an active site which is comprised of an iron hydroxide, ferric hydroxide. The substrate is a long lipophilic molecule with a carboxylate at one end. And what we're going to do is transfer a hydrogen atom out of carbon-11, which is smack in the middle of the molecule, which is then bound to this buried active site to the ferric hydroxide. And this is formally what's called a proton-coupled electron transfer. The proton goes onto the oxygen of the ferric hydroxide, and the electron reduces iron 3 to iron 2. We could call it PCET. It's more like a net hydrogen atom transfer in this system. Okay. Now, let's just focus a little more on the active site. What we see are, very interestingly, a triad of hydrophobic side chains, bulky hydrophobic side chains that are absolutely conserved in the lipoxygenases that have been studied. And here is our reactive C11, the hydrogen atoms going from here to here. Note a bulky residue 754 and another one on the other side, 546. These two are postulated to position the reactive carbon so that it can approach the ferric hydroxide functional group. It's really the active site cofactor. And we also now have another hydrophobic residue conserved. One helix turn away. It's called ILU 553. So by virtue of limited time, I want to give you an example which is the most conservative one. And we're going to look at 553, which is a helix turn away. It's a little remote from the bond that's being cleaved. And all we're going to do is change the size of the side chain. And it starts at ILU. We make valine, leucine, alanine, and glycine in it. The volume is getting smaller and smaller. And what we're doing is introducing packing defects into the center of the protein. Okay. When we do that, what we see is that the rates go down, but not hugely. This is a remote mutation. Rates go down very dramatically the closer we get to the reactive carbon. The enthalpies of activation are pretty small. Around 2 kcals per mole is the largest enthalpy of activation. But what really is changing is the temperature dependence of the kinetic isotope effect. This is exactly what I introduced you to earlier. It's very small. The error is about plus or minus 0.2 here. We're not at 0, but the slopes are very similar in the wild type. And the valine, which is structurally quite similar, actually, to ILU, we've already increased the energy of activation for deuterium relative to protium. So the difference now is 2.6. You go through the whole series. By the time we get to glycine, you can see that the transfer of the deuterium has become more difficult by 5 kcals per mole relative, or subtract the 0.9, 4 to 5 kcals per mole relative to protium. Nothing has changed. All we've done is introduce an isotope. The potential energy surfaces are the same. We have mutations that introduce defects. But this shows that very clearly that this is a tunneling mechanism. I'll go through the model in the next slide. But it's the difference in energy of activation for deuteron versus the proton. And if you think about that, where nothing else is perturbed, it's the wavelength. The wavelength is shorter for deuterium than protium, and therefore when you make these perturbations, the donor acceptor move apart. Protium has enough of a tail, it can get over to the product, but deuterium doesn't do as well. And the way that this happens, leads to these data, is that the enzyme tries to recover because it's got a packing defect. It starts doing this donor acceptor distance sampling, And it has to do it much more for deuterium than for protium. Okay, let's go forward and see what this looks like. This is the worst slide I have, and I think they'll get much simpler after this. But I really want to drive home what the model is that we've arrived at for hydrogen transfer in condensed phase with enzymes in particular that is needed to accommodate what I've been showing you thus far. H transfer, it's a multidimensional process. It's a quantum mechanical process. It's multidimensional. And we write the rate expression, and this is very interesting in terms of Bath reaction coupling. We write the K observed as a product of an equilibration of the enzyme among many different substates, only some of which create the geometries that are needed for reaction to go forward. And then we have our tunneling rate. And this is, of course, what we've been focusing on in the context of Marcus theory. But this is multiplied by a stochastic sampling as the protein vibrates and moves around to reach a small substate of configurations that can support tunneling. And this is shown as Kx in A. We have, in fact, in the system, looked at the ground states. We can show that the ES complex is an equilibrium between many inactive states plus what we call R equilibrium, which is when the donor and acceptor are at van der Waals distance. We also know that that is not short enough to support tunneling, and we then propose this R naught where we go through further conformational sampling to get closer, which is about 2.7 to 2.8 angstroms, which is the distance that most theoreticians will agree upon is required to get functional H tunneling from a donor to acceptor. Now, the tunneling portion of this K observed is Marcus theory. And essentially, what has to happen once all of this is happening at once. I'm breaking it down into the different dimensions to give you an idea physically of what's going on. So, in fact, we also have another coordinate in which we have to get degenerate energy levels. We heard about that extensively in the context of electron transfer. And so what we have are this jiggling of the protein that transiently now generates what we call the tunneling ready state. Two things have happened here. One, we have this transient energetic degeneracy, and two, we've gotten close enough that we have a distance that is almost at the point where it can support wave function overlap between the donor and acceptor. So the temperature dependence of the rate is not the actual wave function overlap, but the activation barrier comes from all of these other processes. That is the native enzyme. Now, why do we see this trend in the temperature dependence of the KIE? That's because with hydrogen tunneling, and maybe also with electron tunneling, we have an additional coordinate, and that's the donor-acceptor distance sampling mode. If all we can do is get to the 3.1 angstrom distance, that's van der Waals, we're never going to see the tunneling. Now, what we propose is that this K equilibrium in the wild type works well enough. We're coordinating the large matrix of the protein with the actual configuration at the active site. So in fact, with just this large-scale global conformational sampling, we get to this tunneling-ready state, which is about 2.7 angstroms. In the wild type, it's almost equally good for protium and deuterium, and that's why there's no difference in energy of activation for the two isotopes. However, once we make mutants, the donor and acceptor move apart. Also, the functional for the donor-acceptor distance sampling gets looser. The force constant, things have loosened up near the active site. The force constant is less. And now we get this attempt of the enzyme to recover the wild-type donor-acceptor distance. It's easier for protium, which is dark purple, than deuterium, which is light purple, because of the tail on the wave function itself. The deuterium is heavier, it's got a shorter wavelength, and it has to move more in order to get to the point where we get our wave function overlap, which is shown here. Is this clear to people? Yeah? Good. Okay. I'm sorry to keep on asking you, but sometimes I have the feeling when I'm talking about this to people who haven't thought about it that, you know, it's just rain coming down, but not necessarily making things wet. Okay. So, relationship to evolved function. There's our model. These data are a sample. We have many systems that show the same results. So, as I said, small temperature dependence of the KIE for wild-type enzymes indicates a close approach of the donor and acceptor. That is, we can get to the tunneling-ready state with no further or very little donor-acceptor distance sampling. This is how proteins evolve. This idea of close approach in protein substrates is generic, I believe, to all of enzyme catalysis. So, how do we do this? The structure of the EAS complexes certainly shows us van der Waals distances, so the key role for dynamics, and these are the global protein dynamics that are bringing us close together in the wild type. Okay. So let me show you one experimental tool we've used to try to get at the nature of these global dynamics and to relate them to the matrix itself. These are very difficult problems because we know proteins are dynamical. We know that they're moving all the time. We know that there are many substates. Many people report on protein dynamics, but our goal is to relate protein dynamics to function, right? We want to know that we can observe changes in protein motion that we can relate directly to the efficacy of catalysis. So one tool we've introduced quite recently is something called hydrogen-deuterium exchange coupled to mass spectrometry. So proteins are moving all the time. You dissolve the protein in D2O instead of H2O. Now the backbone amides will exchange with a certain rate constant. So this is illustrated. We have a large protein. This protein is 94 kilodaltons. And you wait a certain length of time. Deuterium goes into the backbone of the protein. You then stop it by going to low pH and low temperature. You digest with proteases. You get out hundreds of peptides. All right. Then we find the peptides that will map to the protein and give us almost full coverage of the full sequence of the protein. In this case, we get 46 non-overlapping peptides. And we look at the rate of HDX as a function of time, temperature, and most importantly, functionally impactful mutants. All right. Now, there are many things we look at here. These are complicated experiments. We've dedicated a lot of time to figuring out what's going on. And I just don't have the time to go into that here. The most important result is shown here. And what you see is the full visualization of the catalytic domain of lipoxygenase, which is in and of itself about 78 kilodalton. It's very large. And what I'm showing you now is the result of looking at all of these peptides, the peptides that are necessary to give you full coverage of the protein sequence. And the point of this slide is to indicate there are only two peptides, and only two peptides alone, that change with mutation, change their temperature dependence with mutation. So what we have then is the rate of hydrogen-deuterium exchange across the whole protein. We do that over several temperatures. We look at the temperature dependence for the rate constants for exchange. And we find those that are altered by mutations that are known to alter the hydrogen tunneling at the active site. And so the two peptides that we were able to locate are 284 to 299, labeled here, and 317 to 334. They're orange. The active site is located by this also orange ball, which is the active site iron. These, on average, are about 20 angstroms away. Depends on where you pick your residue to focus on. But if we just focus on this loop, 317, 334, it's quite far away from the active site iron. And we look at how the hydrogen-deuterium exchange responds to temperature in the three mutants, and here we have it, the wild type. This is 553G from that first series I showed you, one helix turn away from the active site iron. And then we also mutated 546, which is one of the two residues that sits on either side of the active site carbon. And so the energies of activation for hydrogen-deuterium exchange are altered by active site mutations. And most importantly, when we look at the correlation between the change in the energy for activating this loop for hydrogen-deuterium exchange versus the energy of activation for transferring the hydrogen, there is this beautiful correlation. Now, the actual magnitude of the energies of activation are different. we're looking at different processes. One is this opening and closing of the protein and the deuterium going in from the solvent. That's the energy of activation for hydrogen-deuterium exchange on the y-axis. Catalysis is limited by that complex equation I showed you with the large K equilibrium, lambda, delta G naught, et cetera. But they correlate. This is very significant because this is the sort of thing we've been looking for a long time. This implied to us a network of thermal motions from the solvent interface, which is where this loop is, to the active site. And here it is. Now, remember, this is a huge protein. This is the only region we were able to pick up through this methodology. And here is that loop I mentioned. Here's the iron. Here are the two residues. Well, there's one of them, 546, 754 is back here. The active site, carbon 11, sits in this little valley here, and then the rest of the substrate bends up to the top of the slide. And then here's 553, one helix turn away. And there's this remarkable series of hydrophobic side chains interacting and reaching up to the hydrophobic side chain of 553 and 546 that sits behind the reactive carbon. There's one hydrogen bond in between. We've mutated that. We've seen impacts of mutation, for example, at Y317 that support the role of this connectivity going from the solvent-protein interface into the active site. So this is the sort of thing that is extremely important. This is the bath, everybody. But the bath is not the whole protein. The bath is not isotropic. And these proteins have evolved selected regions in which to now, in our view, promote thermal transfer from the real bath, which is the water, into the active site, which has to undergo this sampling of distances and energetic relationships. Okay. Now, I want to say, how much time do I have? 0? Oh, okay. Then I'll just go through these very quickly. I thought I'd go too slowly. Sorry. Okay. I do need to show you this because this really is the coup de grace in my view. So, outlier. What are outliers good for? Well, either they're good for keeping you up at night and destroying a model, or they're capable of proving a model. And this is, in fact, a double mutant. Everything I've shown you so far is a single mutant. Now, all we're going to do is take the two hydrophobic residues that bracket the reactive carbon. We're going to reduce their side chains from leucine to allene, both of them. We solve the crystal structure. The cavity's much bigger. Okay. Things are moving apart. We measure the kinetics. The hydrogen rate is down 10 to the 4-fold by just reducing the size of two hydrophobic side chains. You then look at deuterium transfer. The isotope effect is in the range of 700. The deuterium transfer is down 10 to the 7-fold. Almost there. We do the temperature dependence. These are very hard experiments with these huge rate reductions. And it's virtually temperature independent. It looks like wild type. But how can the isotope effect be 700? And essentially what's happened, here's the model. We have done many, many biophysical probes to characterize this double mutant. I will summarize it in this simple picture. Essentially what's happened is we have a double mutant. The first mutant, 754, so the blue structure is this is from a QMMM with Sharon Hammett-Schiffer, this ground state model. The blue structure shows the ground state of the substrate where now the carbon 11 here has moved into a pocket that's been created by deletion of 754. In the wild type, the carbon 11 remains further up toward the C terminus of the substrate. Okay, so the reactive carbon is sitting in a different position in the double mutant. It's fallen into a hole that's created by mutation. The other thing that's happened is it now moves this reactive carbon away from the dynamical conduit. So here is the substrate. In the wild type enzyme, it has access to this conduit that leads out to the solvent. But from these biophysical probes, we can show that in the double mutant, this reactive carbon is no longer able to capture these dynamical fluctuations. So the isotope effect is huge because the substrate has moved into a hole. It's gotten trapped there. and it doesn't have access to that dynamical network to get it into the right position. Now, once again, protium is impacted, but deuterium, it hits a huge brick wall. Okay. So I think that I've said most of this, and I don't want to take more time than I already have, except I want to emphasize that I believe these properties are showing us the role of dynamical sampling in order to get very close distances between donor and acceptor atoms. Because hydrogen can tunnel when we get very close, we're now picking up tunneling and with properties that we may not see in condensed phase for model reactions. I think this is a generic property of enzyme reactions, and we're seeing tunneling so often in enzymes with their unique properties because of the way enzymes have evolved to catalyze all the chemical transformations that they have to do. I did have another slide. Oh, yes. Question for my audience. With the double mutant of SLO, note that even when tunneling has been very impaired, we do not revert to classical behavior. This is not the behavior anticipated for a competition between classical and quantum mechanical behavior. It's pure quantum mechanics. Okay. I don't have time for this. But if people want, I'll discuss it later. I really did want to have a chance to talk about proton tunneling or supposed proton tunneling in DNA. So perhaps the organizers had asked me if I could give a longer talk in my own defense last night when I heard that the second speaker wasn't available. Should I just bypass this, or what do you think?
I think we should move on to
the next speaker. All right. It's too bad. But okay.
Sorry.
Many, many people. And I want to point out also that nobody would touch this with a 10-foot pole many years ago. The only money we could get to study this project came from the National Science Foundation. And in subsequent years, we were able to get large amounts of money, especially from the NIH. But I'm indebted to the NSF, and I wanted to point that out. Thank you very much.
Time for one question while we set up the next speaker.
Does anybody try to apply pressure in order to bring the Broglie wavelength together and see the activity of the enzymes, for example?
Yeah, we've applied pressure. There are other groups that have applied pressure as well. The temperature dependence of the KIE, sometimes it varies, sometimes it doesn't, but the thing that's affected the most in our hands is the energy of activation. Essentially, what you're doing, protein, it has, in this case, a protected active site, and the matrix is much floppier. So when you apply high pressure, you may be ordering local electrostatics, hydrogen bonding, and now this conformational sampling, which is so relevant, becomes harder and the energy of activation goes up. So to just squish the active site is very, very difficult, but you certainly impair the enzyme in ways that alter the distribution of protein substrates.